Good morning, everyone. I, Ritika Saxena, Assistant Professor in School of Biotechnology, IFTM University, Muradabad, is here to deliver a lecture on Human Genome Project. Human Genome Project, a project that actually deciphered or decoded the human life. I will be delivering lecture on the following content: a brief introduction about the topic, the goals of the project, the pathfinders in the project, participating agencies and countries, landmarks or milestones in the project, sequencing strategy adopted by the project, that is hierarchical shotgun sequencing method, a parallel project run by Celera Genomics, and finally, I will be discussing the outcomes and future applications of the project. Human Genome Project. It was a 13-year integrated effort of the United States Department of Energy and National Institute of Health. It started in 1990 and was finished in 2003. It planned to determine the sequence of human DNA and also mapping all the human genes. Till date, HGP is the world's largest collaborative biological project. The project aimed at the following: to get an idea of all the genes in human DNA, to figure out the sequence of 3 billion nucleotide bases that make up the DNA, to attain a physical and linkage map of the genome, to maintain inventory of this information in databases, that is bioinformatics, to improve the infrastructure of human genetics, and to address the ethical, legal, and social issues related with the project. Talking about the persons that led to the path of this project, good science can only happen with good scientists. So, in the complete project, a remarkable collection of scientists representing many countries, many different disciplines, and many levels of seniority coalesced around these shared goals. Robert Chinchmer, Chancellor of University of California, gave the first proposal to start the project in 1985. Charles Delphi, Director of the Office of Health and Environmental Research of U.S. Department of Energy, decided to begin funding the research into genome mapping and sequencing in 1986. In 1988, a special committee, National Research Council of the U.S. National Academy of Sciences, recommended the initiation of Human Genome Project. The project received a significant boost in 1988 when Nobel laureate James Watson commented that only once could I have the opportunity to let my scientific life encompass the path from double helix to 3 billion steps of human genome. He headed the joint venture of NIH and DOE in 1988. In 1993, Francis Collins succeeded James Watson after his death as the project head and director of NIH and completed the project in 2003. Now, the accompanying agencies and countries: U.S. Department of Energy and National Institute of Health initiated the project in 1988. Government-funded sequencing was operated all around the world in the United States, the United Kingdom, France, Germany, Japan, Great Britain, and China. Now, the landmarks or milestones that the project achieved. In 1986, the birth of Human Genome Project took place. In 1990, the project was initiated by DOE and NIH. In June 2000, an initial rough draft of the human genome was available. By February 2001, a working draft was completed and published, and it was declared completed 2 years ahead of the schedule in 2003. A major quality evaluation of the human genome sequence was published in May 2004. Now, the sequencing strategies that were adopted by the project. HGP followed the hierarchical shotgun sequencing method. It is also known as back-to-back sequencing, clone-by-clone strategy or even top-down sequencing. It is done by mapping first and sequencing later, and the technique was more accurate at that time than any other technique, but was time-consuming and costly. In the hierarchical shotgun sequencing method, the genomic DNA is sliced into fragments that are approximately 150 MB in size and inserted into the back vector. These vectors are transformed into the E. coli cells where they are replicated and gathered. The back inserts are confined and charted to determine the order of each clone's 150 MB fragment. This step is known as the golden filling path. Each fragment in the golden path is disintegrated randomly into shorter quantity, and each piece is cloned into a plasmid and sequenced on both strands. Alignment of the sequences is done so that the equivalent sequences are found overlapping. After each strand has been sequenced, the contiguous pieces are then masked into finished sequences to produce a 8x coverage of the high quality data. This was the technique or method adopted by the Human Genome Project. Together with the project led by the government, a parallel project was being run by a private company named Celra Genomics, whose owner was Dr. Craig Winter. Celra proclaimed that it would sequence the human genome within 3 years. Celra developed and adopted the whole genome shotgun sequencing method for sequencing the human genome. The shotgun sequencing constantly scissors the genomic DNA into tiny fragments, which are duplicated into plasmids and sequenced on both strands. Once these sequences are obtained, they are aligned into finished sequences. This technique eliminates the back step from the HGP's approach. It is less expensive and quicker than HGP's technique, but it is more susceptible to error due to the incorrect assembly of the finished sequences. Now moving on to the outcomes of HGP that the project achieved. The project aimed to create a genetic map of 2 to 5 centimorgan resolution map, whereas it actually achieved a 1 centimorgan resolution map by September 1994. It also created a physical map of the human genome that showed around 52,000 of sequence tag sites by October 1998. The complete DNA was sequenced with an accuracy of 99.99% by April 2003. 3.7 million human SNPs were mapped by February 2003. Great achievements were obtained with the project. Now moving on to the current and future applications of HGP. One of the major applications of the project is in the field of molecular medicine. With the help of this research, medicine will look more into fundamental causes of disease rather than concentrating on treating the symptoms. So, it will lead us to a better diagnosis of the disease. It will help us to detect the genetic predisposition to disease, enable the geneticist to identify transporter within families, and also able to construct curative products based on new classes of drugs, immunotherapy techniques, and feasible restoration of faulty genes through gene therapy. Another major application of this project was in the field of biotechnology. It popularized the expansion of new biotechnology companies anticipating to take advantage of the significance of its research. The project also has its application in the field of evolution, where we can study evolution through germline mutation in lineages. It, it also makes us able to compare the breakpoints in the development of alteration with the ages of population and historical episodes. Understanding the human genome will have an enormous impact on the ability to assess risk posed to individuals by environmental exposure to toxic agents. And it will also give us improved consideration of biology, increased taxonomic understanding, heightened improvement of pest resistance and productive crops and livestock, and other commercially advantageous microorganisms. Moving on to the conclusion and future prospects of the project, it made us understand about the human origin. It also led to the emergence of various fields like system biology, bioinformatics, proteomics, genomics, immunoinformatics. The project also deepened the concept of personalized medicine and pharmacogenesis. These are the references for the lecture that I prepared. Thank you. I hope my lecture will provide you a deep insight into the topic. At the end, I would like to say that though HGP was completed in 2003, it still has a long way to reach its ultimate goal.